Today we get to take a journey. We are going to be a product of the cell. We're going to talk about how the organelles you've already learned about, make sure you've done that assignment first, how they work together to create things, to move things, to make things happen in the cell. So we're going to be a product of the cell. We are going to be something the cell makes. And so we're going to start with one we know best. We're going to start with a protein. So we know that proteins begin in the nucleus because that's where the DNA code is. But they're not made there. You know that they go from the nucleus, the mRNA goes from the nucleus into the ribosome, where makes a lovely amino acid chain. Once that amino acid chain is formed, what happens to it? Well, remember, we mentioned briefly in class once that you have two options for ribosomes. You have ribosomes that are bound to the endoplasmic reticulum, and you have ribosomes that are free, floating in the cytoplasm. So it depends on what kind of protein you need to which ribosome is chosen. We're going to go with the one where you are making a protein that will be excreted from the cell. So remember that we used lactase as an example, and so we had a lac operon that created the mRNA at the right time because we had lactose present in the cell, and so we needed lactase, right? So now, but lactase actually is usually it's usually used outside of the cell, or it can be. It needs to be because that's where the lactose, the bulk of it is. And so what will happen then is you need to get that lactase made, and you need to get it out of the cell into the digestive tract where it can break down the lactoses there. So how do you do that? Well, if you're a cell, first of all, you're going to make that particular enzyme on a bound ribosome, one that is attached to the endoplasmic reticulum. And so because it has a certain pathway it needs to travel, so it can't just be made on a free one, because the free ribosomes usually make things that stay in the cell or are used by the cell. And so we're actually on a bound ribosome. So we're in a ribosome that is attached firmly to the wall of the endoplasmic reticulum. That's where we're going to be made because we're a lactase enzyme. And then we are going to move through the endoplasmic reticulum. And while we're moving, we are going to be folded as a protein. Remember, our protein is an amino acid chain, right? So it's a long strand of amino acids. So it's going to be folded and modified and maybe put with some other proteins and made into this nice package. And when we get to the end of the endoplasmic reticulum, there'll be a little vesicle that pops off. Isn't that nice? The vesicle is a little sac that allows something to be carried through the cell and to a specific destination. You can't just have things floating around all willy-nilly in the cell. Like, that's not good. They need to be regulated. And so then your vesicle is going to go to the Golgi. Why? Because I love the Golgi. The Golgi is the packing and shipping place. So what do we have? We have lactase. Where does it need to go? It needs to leave the cell. Who's going to tell it how to get there? The Golgi. The Golgi is going to process and tag it and put it in a different vesicle. So the Golgi is like a membranous hallway, if you will, where the vesicle from the endoplasmic reticulum can fuse. Notice all these membranes are the same, so they can just fuse together. So it fuses into the Golgi, and then the Golgi goes through, the little enzyme goes through the Golgi's hallways, and in the process, it's tagged so that it can be sorted and go to the right place. Okay, so now we're at the end of the Golgi, and it pop off a little vesicle, and the vesicle is going to go to the cell membrane, and because it's a membrane, it can just fuse to the cell membrane and the lactase out into the cell where it can be picked up by the bloodstream or whatever it needs to be, and it can be taken to where it needs to go. Okay, so that would be how a protein is created that is secreted from the cell. Now there's other ones that are not. So if you need it actually in the cell, then you would just go to a free ribosome. It would be made and packaged and taken by motor proteins to where it needs to go. Could happen that way. What if stuff needs to be broken down? Well, then it would be carried. So if a cell engulfs something bad, because maybe it's an immune cell, maybe it's engulfs something bad, it can be carried to the lysosome, and the lysosome can take care of it. So all of these organelles work together. Like the membrane allows stuff in, and then it's transported by transport proteins to where it needs to go. Most things are going to be like When it comes into the cell, it's going to be wrapped in like a vesicle. And so that vesicle will carry stuff out. And so you need to be able to think through it like that. Like we don't just need to know, for pity's sake, the mitochondria is a powerhouse of the cell. Like that's the most ridiculous thing, because then students think that they know it all, but you don't. You need to actually be able to put them together into groupings and say, this is how they work together. And so that's going to be your job as you grow to be AP biology students. I hope that helps you. Think it through. Have a great day, and I'll see you soon.